16. Another important topic related to protein modeling is protein dynamics, and even if they are old methods that have been applied since 1970s, they are not totally obsolete because the dynamic nature of proteins is something that the best modeling procedures can't really touch. AlphaFold 2 only gives you one protein model, so it's like a snapshot of the protein at some point of its real dynamic life cycle. And it's really important to remember that proteins are not static, so there's a lot of movement happening at different time scales. And you might consider proteins as elastic material, so they take some stretching, they take some compression, and they return to their original form. There's a large amount of small, weak molecular interactions like hydrogen bonds and van der Waals forces, electrostatic interactions, etc., that hold the proteins together. And they are like your Velcro material, the stuff that puts your rucksack straps or whatever. I think I have a little bit here. So this strap around the cord of my power cord of my laptop does have the Velcro. So even if the things are really small, so they hold this thing together because there are hundreds of them. Each individual tiny plastic hook would be negligible and couldn't really hold anything wrapped around the cord, but with 1 square centimeter that is really strong. And in space stations the astronauts have something like 20 by 20 centimeter patches of Velcro in their clothing, which they stick into the counterpart on the wall, and in that way they can sleep anchored to the wall without being afraid that the movements of the space station would tumble them across the walls. But I'm digressing. So proteins are elastic material because of a large amount of weak forces, so that you can break a few of them and they will tend to reform when the protein finds its native conformation, provided that the forces are not so large so that the protein is denatured, it's losing its native fold and maybe aggregating or misfolding or whatever. And these weak forces also allow for changes of conformation. We have done some morphing of a single protein, which clearly shows that we can crystallize the protein in two different forms with phosphorylation or without phosphorylation maybe, and then the electrostatic interactions provided by the negative phosphate groups might be enough to trigger some pretty big movements. Or in case of hemoglobin, when we have oxygen binding, it is pulling the iron back to the plane of the heme which is slightly bent in the deoxy form will be straightened, and then that will move many other parts in the protein. So we can have these conformational changes, and the difference between a chemist and a biochemist is the understanding of proteins. So if somebody is asking how the enzyme works, well, it's these conformational changes in the protein. It's kind of a blanket answer that doesn't really explain the details, but it gives you the fundamental reason, in the same way as a physicist can fool a chemist, or let's say a chemist can fool a biochemist when they are asking about a reaction mechanism. The chemist can say it's a quantum mechanical thing. Okay, and then the questions are stopped. Well, anyway, so we have these conformational changes that are essential really for enzymatic reactions to happen or for receptors to be activated. When we have a ligand binding to the outside of the receptor, so here comes the ligand, so there is some tiny movements in the amino acids which are binding to the residue, and they lead to more changes that are transmitted even across a cell membrane to the inside of the cell, making the cell realize that, okay, this receptor is occupied, and then something happens in response to the ligand binding. It could be a hormone, and that could lead to a change in the gene expression, etc. So all the wonders of biochemistry are driven by conformational changes. So it's important that you understand the dynamics. And as a good example, here's an enzyme that consists of two domains, and when the substrate is binding, so the substrate is shown here, it is kind of a phenol derivative, and so then the domains of the protein are folding like the jaws of Pac-Man to enclose the substrate. Many channel proteins have movements of alpha helices in the membrane to have the protein open to the outside, closing the glucose, and maybe opening to the inside to let it go through. So all kinds of changes are happening. And here's another extreme example of proteins called integrins that have an I domain. These integrins might be sitting in a bent and folded conformation against the cell membrane, not being reactive for their ligands, and when the cell is activated in somehow, this conformation is then straightening, and that allows contact of this I domain for another molecule ICAM1, which might be on the surface of another cell. So we have all these large-scale movements, and it would be so nice to understand them by computer simulations, because obviously there are only so many crystal structures you can create from the same protein. And things that are large as this might be too hard to crystallize, and maybe they never get these conformations if you try to detach them from the cell. And maybe you could do something with cryo-EM, but again, that might be just a single conformation. With luck, you could have some particles that are folded and others that are straightened up. But anyway, these are huge differences that do movements that are in scale of several nanometers. The cell membrane is maybe 4, or is it 2 nanometers across, more or less 20 angstroms, or maybe more. Is it more like 30? Well, anyway, it's in the order of a few nanometers. So when this is standing up, it is dozens of nanometers, or hundreds of angstroms, if you wish. Individual protein domains are usually between 50 to 100 angstroms. So what are the methods that you can use for studying protein dynamics? We can do experiments in many physical methods, spectroscopy like NMR and crystallography. They do work if we have access to multiple forms of the protein. There are some light scattering methods that give you an idea of the general shape of a protein particle whether it's globular or whether it is more like a long rod. These are like a start in understanding protein shape, and maybe you can trigger the change between a globular and a rod-like form by changing the pH or adding some ligands or whatever. Then there's atomic force microscopy, which is a method of studying surfaces at a kind of a crude resolution, and then cryo-EM, which is also a method much developed. I would say that this AFM is being left behind, and electron microscopy is the method of choice for observing protein-sized particles directly. But then for computer simulations, you can do quantum molecular mechanics for small molecules, and then for proteins and DNA, molecular dynamics is the way for observing changes or possible changes in a protein. So in molecular dynamics simulations, what you try to create is a model of molecular movement. You're artificially heating the molecule so that there are all kinds of molecular vibrations and maybe some further movements there. And what you need there is an initial structure, maybe an X-ray model or a computational model from AlphaFold or whatever. And then you need to define the physics that make the rules that the protein movements are based on. And actually this is very similar to molecular dynamics in that we can use the same force fields or very similar force fields for figuring out the forces on each atom there. And then you need some physics for making the particles or the atoms move. Initially, you can't do much dynamics if your protein is frozen at absolute zero Kelvin. That means that no atom is moving anywhere, and that is what you have in your initial model. So you have to give these atoms some distribution of velocities, which should be matching kind of velocities that the atoms would have in a real protein. So you're assuming maybe Boltzmann distribution for different velocities, and I have a bit more to say about that in a later slide. But still, the physics for the forces affecting the atoms is coming from 1950s and 60s. It is the Newtonian physics simplification of atoms into point-like objects with tiny masses, which would be moving exactly with classical schoolbook physics, nothing fancy or quantum mechanical there. And we had a reference here, 1977. That was the very first protein molecular dynamics simulation. It took a while to have computers as big as you need for computing hundreds of or even 1,000 atoms. This was a tiny protein, bovine trypsin inhibitor, bovine pancreatic trypsin inhibitor. That's the protein. I think that's about 60 amino acids in size, and that would be maybe 600, 700 atoms, or maybe more than 1,000, including the hydrogens. But anyway, for the simulation, this is kind of important, especially the idea of going through time steps of very short times, like 1 femtosecond for the movement. So I'll try to go slowly here. what's happening. So for starting, you need to have the initial coordinates. You need to have your protein complete with hydrogen so that it's being prepared. You need your force field so that you can start calculating the forces that affect each atom. And then at the starting point, you give some random velocities to the atoms, and the distribution of the velocities would be by the Boltzmann distribution, and then the magnitudes of the velocities can be calculated to match some simulated temperature. And actually, it's commonplace that you heat the proteins at awful temperatures that wouldn't be in any way possible for real proteins. They would be denatured, but because the simulation is so short, the protein doesn't have time for falling apart, and because you stop it every 1/femtosecond, the movements are short, so that the proteins are not really breaking apart. You let the directions of the particle change after each time step. But anyway, you give these random velocities, and then you have to balance these velocities so that the sum of all the velocity vectors is zero, so that you match the y, the x, and the z components of all the velocities so that they sum to zero, because otherwise when you start the simulation, if there are more particles that are moving to the right than to the left, it would mean that your protein starts running to the right, and after a few steps of the simulation you would lose it outside of the screen, and after hundreds of steps you don't really know if it's going to be on the next street or the street after that. So you need to make sure that the protein stays in the same place. And in the similar way, you have to balance the angular momentum of rotating the protein, because if there's an unbalance, the protein will start spinning in an accelerating fashion. You have to have a balance between the momentums in each direction. But anyway, this is probably more details than you wanted to know, but I still wanted to tell it because it's such a cool idea that you would have your protein that is accelerating and making it escape from the screen, and then you never see it again. So it's much easier if it stays in a nice coordinate system that might be the size of the unit cell of a crystallographical experiment. But anyway, once you give these initial velocities to each particle, you let the proteins move in the direction of the velocities for 1/femtosecond, 10 to the minus 15 second. And that is a short enough time so that you don't break any bonds. If you do, let's say 1 picosecond, so it would mean that if your hydrogen is moving away from the carbon, it would be just so far away that there wouldn't be any chemical bond to speak of, and the force created by stretching the bond 10 times its length or 100 times of its length, that would be so much that in the next time step the hydrogen would come back and go through the protein and wreak havoc. So you need to be modest and let the atoms move only for a tiny distance at a time. But anyway, you can calculate, once you know the velocity and the time, you can calculate the distance the particle travels, and then you have a new set of coordinates for your protein. And then the forces will be now different. You assume that there are no forces in the starting situation, but now with the movements, there will be some bonds that are not in their ideal length. There will be clashes, there will be bond angles that want to come back. So then you start calculating new forces and new velocities based. You start calculating new velocities based on the forces that are affecting these particles. They are giving them acceleration into other directions. And then you let your atoms move for another time step. Again, recalculate the forces and recalculate the velocities, recalculate positions, and you iterate this over millions of times. And it's typical that you save one snapshot of your protein every 1000 steps. There's no point in trying to save millions or billions of protein structures because the changes will be so minimal, but taking one sample every 1000 steps is good enough to create a view of how your simulation is going. And what you get is a trajectory for each atom, or you can talk about the trajectory for the collection of atoms. It would be the time course of the movement for each atom, in a way that when you throw a ball, the trajectory is the curve is doing over time, but in this case the trajectories would be going pretty much back and forth. And maybe with luck you can have some loop changing its conformation so that the trajectory could lead into some actual net movement and not only staying in one place. And these trajectories are the stuff that the molecular dynamics guy will be analyzing to see if there is something strange like one atom changing its place so that there is a kind of a balance of the protein staying somewhere, but after some energy barrier has been crossed because there's movement, it can happen, maybe relaxing into a new equilibrium position, there will be a new position for that atom or several atoms in a protein loop that will look different. So these things can be found either visually when you are watching a movie of the trajectory, or maybe you can do them computationally so that you observe something like looking at the bond length of a specific amino acid to a ligand, or the distance between the ligand and one amino acid to kind of formalize or make a graph of the specific interaction to see how it changes over the simulation. So it's kind of complicated, but I hope this explanation is, I'm not going to go any more technical than this. So I hope this makes you appreciate the idea of how you can create a simulation over time using the same kind of approach as you have for molecular mechanics for energy minimization. And now for the time scales, now this is small print. I need to go to my own copy and watch it here. So the unfortunate thing in the time scales is that we can only reach the high microsecond scale in this molecular dynamic simulation. With luck, you might reach even a couple of milliseconds, but that would require months and months of computer time. Whereas the actual movements of proteins are longer than that, so you can't really simulate the movement of a hemoglobin, the movement of domains across each other, but you may simulate some loop motions and changes in the positions of side chains, the side chain rotor mass. These are in the scale you can do. Then the limitations here is that's mainly computational. We can't increase the time step too much because the fastest vibrations for carbons and hydrogens, carbon-hydrogen bonds, are happening in that time scale. You can't let them run. You can't let those bonds to be stretching in one direction. You have to stop them. And by the way, the temperatures that the simulations do for proteins are typically 600 Kelvin, so that would be almost 300 degrees Celsius. So it would be too slow if you leave the protein at 300 Kelvin. So 600 Kelvin is actually more than 300 Celsius. Okay, so because of this time scale, you can't really do these slow movements, and having the diffusion of a ligand into the protein, it's again too slow. You have to start with the ligand in place, and then you may see how the shape of the ligand is changing when it's bound in the protein, and the specific distances to certain amino acids might be changing. Okay, the time steps can be lengthened if you give up the atomic approach. You can do coarse grain force fields so that instead of letting individual atoms move, you make lumps of 4 to 5 atoms, which are then the particles that are moving. And then you can do time steps of 20 to 40 femtoseconds. And you might also force movement into a protein. So if you want to study the straightening of your integrin, you can create an artificial force which is pulling from the two ends of the bent version of the molecule, and when you pull them apart, it will force the protein to be straightened. And if you go on with the stretching, you might be able to see which is the weakest link in the protein, which is the domain, which is going to be ripping apart, which is going to be ripped apart when you have the force there. And then you can't really do huge proteins, again, because of computational limits. So you can simulate maybe 1 million atoms, and if you want to do realistic simulations of protein, you probably want to have them in water instead of vacuum, and then most of the particles in your system are going to be water molecules if you simulate them as explicit water molecules. You can do things like applying a blanket dielectric constant, which is different from the dielectric constant of the vacuum, so that you are decreasing the range of electrostatic forces as it would happen in water. But anyway, if you are using explicit water, you have to take care of not losing your water. If your system is a cube of water and then there is vacuum all around it, the water would be just running to all possible directions, and you would lose the idea of having your protein soaked in water. So there is a system of using a periodic system. You can use a periodic system meaning that instead of having just that one cube with the protein and the water, you create a system of 3 by 3 by 3, that is 27 copies of, or maybe you can use just 7 copies, so that you have copies of the cube at the back and front, left and right, and top and bottom of your system. And what happens is that whenever water is going, you keep these systems as copies of each other, and all the movements are copied. And when you have water molecules which is crossing out of the box, so at the same time you have another copy of of water which is coming back into the system from the box below. So in that way you have the same number of water molecules in the box all the time when you have this system of copies. And if you don't have the box around the water, so it would be easy to understand that when you start the simulation, so all the water will just drop out of the box, and then you will have a wet keyboard and table. So what can you do? But seriously, so these water simulations are really requiring a lot of computation. And you may also simulate membrane proteins, where you have to create a square of the membrane lipid bilayer. And again, it's probably going to be periodic, so that you have identical patches around with the movement of the lipids being replicated, so that you are not losing anything. And with each lipid consisting of 50 atoms maybe, so it's a lot of computation there. And then there are some further limitations, so you can't simulate reactions. So if you have an enzyme, the reaction is never going to happen. You can't break bonds or create bonds in classic MD. And you have to do kind of quantum chemical things, and maybe be limited to just a small system of a few hundred atoms around your enzymatic substrate. And you can't really change the charge distributions. You might start with a molecule where you do the calculation of charges, so that you can have something else than just the elementary charges or multiples of elementary charges on specific atoms, so that you can have, let's say, a distributed charge in a carboxylic acid, so both of them have approximately half a charge. But that is going to be static, so during the simulation these charges are not changing. Okay, I'll skip the idea here. For the molecule, you have to have the atomic coordinates, and you also have to have a way of knowing which of the atoms are bonded. And with protein specific and DNA specific programs, it's pretty simple. The connectivities have been programmed, and there are libraries that understand amino acids and nucleotides. They know which atoms are connected. But for anything which is not standard amino acids, you have to define the connectivities for let's say a drug molecule which is bound in the protein. And then you need to have your force field, which is able to handle all the atom and bond types which are in your system. and I'll skip the idea of analysis tool, but I'll just mention that you can use the chord set tools and the same tools we use for morphing movies. You can use them for playback of molecular dynamics trajectory, but that is mainly for visualization, and the calculation tools are not as good as in dedicated molecular dynamics packages. And once you have your trajectory, there's a lot of things you can analyze from it. And again, this is going beyond this course. And if you need to look at this later, you have the lecture notes to consult. And finally, we are at the point where we can watch a video. So first of all, let me check if